Scientists work for the money of the people. We all work from taxes that are given us by various governments. We get, at the CRG specifically, we get money from the local Catalan government. We get money from grants from the Spanish national government. We get money from the European Union. I also get money from around the world. So this is a chance for people to actually understand where the money that they give us goes to. So this is kind of a global financial reason. A global scientific reason is because this is really the chance to find out what is happening in science. There's a very big problem of having a science journalist being an intermediate between scientists and the people. Usually when the journalist comes to talk to you, they want a very important line in the newspaper saying, you have cured cancer, we're done. That's never the case. If you were to look at newspapers, we have cured cancer 1,000 times by now. It's much more complicated. It's much more difficult for us as scientists to explain it to the people through somebody else. It's much easier to do it just directly by yourself when people have the direct chance to ask you questions.
I will try to accomplish two things today. I will tell you a little bit about what my laboratory does, and I will try to explain what kind of consequences our work at the laboratory has for personalized medicine, or at least for what people perceive to be personalized medicine currently. This is a small, very small piece of DNA. You might see that there are two strands in this. One is this strand in red, another strand in blue. This is the double helix that everybody keeps talking about since Watson and Crick discovered that this is the structure of DNA. There's one thing, two things actually, that I want you to get from this picture. One is that the structure of DNA is just a simple molecule, right? This is just a molecule. It's just a very big and very, very complex molecule. That's it. The second important point from this picture that I want you to get is that this molecule has a structure. Basically is a three-dimensional shape and space. This is the basis of the DNA. The reason why this DNA molecule is important is because all information that is necessary to make us as individuals actually is encoded in this molecule. And this molecule is actually packed very tightly in the nucleus of the cell. This is actually an electron photograph of an actual chromosome. It's just packed, just ready to be split, right? Two chromosomes into two different cells, and you can actually see this with a microscope, right? What we mean when we say information is encoded, we really mean this very, very simple pathway as we go from DNA into protein. And all it says is from DNA, in order for information to become useful, you first need to read it. And the way you read it is you actually change it into an RNA molecule. Then from this mRNA molecule, you can actually read it again and make a protein. This is a representation of a DNA molecule being read by some kind of a protein and is made into an RNA molecule. Then this molecule has to leave the nucleus. Afterwards it gets to this complex, which is called the ribosome. It's a complicated complex, including RNA and protein molecules, that reads this information from the mRNA and makes the protein. You can see that individual amino acids, and amino acids make up a protein, are being brought into the ribosome by these guys, the guys in blue. These guys are called transport or tRNAs, and their only function, right, is to bring the right amino acid into the right place on the mRNA. You can make this picture as complicated as you want. You cannot physically even. If you were to put all of the human knowledge, even of this simple process, onto one slide, it would not be comprehensible. So what usually people do, and this is usually what we do also in our laboratory, is study specific molecules within this pathway. This is a tRNA molecule. This is its tertiary structure. The tertiary structure is the three-dimensional structure of any particular molecule, right? But the amazing thing about this tRNA is that it also has a different level of structure called secondary structure. What's important here is that this is the secondary structure that allows this molecule to maintain its function. You have this stem structure, which is exactly the same concept as a double helix in a DNA. This is, let's say, ignoring the rest of this molecule here. This is strand in red color that I've showed you before. This is strand in blue color before, and you have hydrogen bonds forming between the relevant amino acids. The cute thing about it is that this is a double helix structure that forms on the same molecule. Here is a representation of how many hydrogen bonds you actually form between different nucleotides. Between C and G, you form 3 nucleotides. Between U and A, you form 2 hydrogen bonds. Between G and U, you actually also form 1 hydrogen bond. Between C's and A's, you don't form any hydrogen bonds. Between all other combinations, pairwise combinations of nucleotides, you also don't form any hydrogen bonds. So given the right combination of these nucleotides here and here, you can have enough hydrogen bonds to keep the structure very strong and to keep the structure stable. Take a look at this case right here. In red, this letter in red, is a case of a mutation that if you were substitute G to A, it would disrupt this structure enough that it would cause a disease in human beings. The reason why this mutation is bad, the reason why it causes disease, is it because it disrupts these hydrogen bonds and weakens the double helix structure of this tRNA. We can start to understand the evolution of such cases where other organisms, sometimes very closely related organisms to humans, have the disease mutations without any ill effects. This is the structure of the chimp. Chimp is our closest living relative, right? And again, if you look at exactly the same position in exactly the same tRNA, the chimp has the disease state without absolutely any ill effects. The chimp is not sick because you have the other interacting nucleotide here. It essentially restores the stability of this structure here. In a human, you have 3 hydrogen bonds here, you're okay. You have a disease if you break these 3 hydrogen bonds. In the chimp, you are okay because you form 2 hydrogen bonds. This protein is a hemoglobin, and you might know that hemoglobin, what it does is that it brings oxygen from the lungs into other body parts. These coils right here is a representation of the secondary structure of this part of the protein. These ribbons here is what the secondary structure of a protein looks like in 3 dimensions. You see that there are these heme molecules here. These are the ones that catch actually the oxygen. It just has 4 parts that are identical within itself. It's essentially a molecule that's made out of 4 bricks that are exactly the same shape, exactly the same structure. Here's another example of a disease mutation. This is the way mutations are written in sort of scientific notation. If you change V at position number 20 into an E, that's a mutation. So the important thing here, right, is that it's not a specific mutation that causes the disease. Right? What causes disease is the wrong interaction of this mutation with some other state. This is called a multiple alignment because we are aligning many, many different species. A capital letter with the red square here means that if this amino acid was in a human, it would cause disease. This is how to read these structures. In this purple color here is what you would find in a healthy human. In red color is the mutation that would be deleterious in human. In blue here is the interaction in the species that I'm showing, in this case the chimp, that actually takes away the pathogenic effect of this disease mutation. I'm just going to skip it. In whales it also happens. In echidnas it happens with different types of methods, very complicated and so on. This is a picture where people have introduced into a bacterial protein a bad mutation, and then they try to introduce other mutations and see if they can recover the normal function of this protein. And they found 3. So 3 different mutations can recover the deleterious effect of this one in different positions. People are already getting data that it's also true for proteins, that the compensation of a deleterious effect of one amino acid mutation can also be possible by the interaction of completely different positions. From this point on, you have to take me more critically, because the data I'll be showing you now is not published, so therefore it has the chance to be completely wrong. This is our familiar amino acid sequence alignment, and this is an alignment of a different protein. This protein is called alpha-synuclein, and mutations in this protein cause Parkinson's disease. The mutation that they found is in this position right here. In humans at site 53, normally there's an A. In the disease state, the amino acid is a T. For example, this rat position right here also has the disease state. They have not been able to discover why this rat is actually completely normal, and this is exactly what we've been able to do. The way we've been able to do this is to look at the evolution of this protein. This is a phylogenetic tree. This essentially tells you who is related to whom and how much. So these are other monkeys. These are old world monkeys. These are new world monkeys. And what I have done here is I give you the position number 53 in alpha-synuclein, and which amino acid is found in this position in these different species, right? Because we know that the common ancestor of all these guys is here, using this information, we can reconstruct the ancestral position to all of these divergences, right? It's clear that the common ancestor of human and orangutan also had an A in exactly the same position. However, at some point here, we know that the ancestor also must have had a T. It means that the disease state can actually be normal to our ancestors. Very, very simple theory tells us that this is the mutation that happened somewhere in our ancestor here, right? They are common ancestor between us and the old world monkeys. So there was a T to A substitution that used to be T, now it became an A. Probably the reason why the ancestor is not sick is because it's interacting with some other position that made this T position bad. So somewhere along this lineage here, there must have been another compensatory substitution that made T worse than A. This is the same representation on the phylogeny of the site of alpha-synuclein. You have an A in the old world monkeys, you have a T in the new world monkeys. We found another protein where people have hypothesized that this protein interacts with alpha-synuclein, where the amino acids have exactly the same phylogeny. So in all of the old world monkeys, there's an S, in all of the new world monkeys, there's a P. So we can make some predictions. What happens in Parkinson's disease is that neural cells, they start accumulating alpha-synuclein, this protein. Normally when proteins are produced, including alpha-synuclein, they get old and they need to be degraded. They need to be broken apart and new ones need to be made. So what happens in Parkinson's is that alpha-synuclein is not degraded and it keeps accumulating in cells. So we can make predictions that if you take absolutely normal human cells and you take regular human alpha-synuclein with A in site 53, and you take the normal regular LAMP2 with S at site 94, you will have regular behavior, meaning that your alpha-synuclein will be degraded with time. Then what you predict is that if you introduce the disease mutation in this state, alpha-synuclein will not be degraded. So our prediction that if you take, again, the disease state of alpha-synuclein and introduce what we think is a compensatory mutation in the other protein, you would restore the wild type. These are our results. This blue guy here is the normal human with the normal alpha-synuclein and the normal other interacting protein. We overexpress alpha-synuclein, and you see that with time, the amount of this alpha-synuclein declines. In red is the introduction of just one mutation, right? If you introduce the disease mutation, actually the amount of alpha-synuclein from this position even goes up. Let's look at this guy right here. So it has the disease mutation, this is a cell line, right? And it has what we predict to be the compensatory mutation, right? So the alpha-synuclein is degraded with time. This is the control, let's say, experiment, right? Where we introduce just the compensatory mutation, the regular wild type alpha-synuclein guy, and introducing just what we think is the interaction mutation, and you actually also get a bad state. You actually also get the accumulation of alpha-synuclein. The reason why I'm showing you this is that if this is true, again, it's not published, if this is true, this will be the first example, experimentally confirmed example of proved compensatory interactions between two different proteins. This is the end of data. Everything else I'll tell you, you cannot even tell your friends because it's completely not true. Speculation is the following. I've shown you that this is true if you consider different species. Nobody has a clue whether or not it is true if you consider different humans. The reason it's not true, it's not necessarily because it doesn't exist between different humans, but because nobody has ever really looked. There are specific reasons why I think that it must be present in the human population, because there's a thing that's called penetrance for different disease mutations. Penetrance is how likely you will be sick if you have a particular mutation. Some mutations are a certain percentage penetrant, let's say 50% penetrant. This means that only 50% of the people who actually have the mutation have the disease. What you would regularly get from a newspaper of what personalized medicine is that if you go to a doctor and you tell him what is the sequencer of your genome, blah, 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 the doctor will be able to look at the genome and say, aha, you have this mutation, and this mutation predisposes you to some particular disease. If I'm right, that's not enough. What the doctor should be able to tell you is that you come and say, oh, this is my genome. The doctor will say, okay, this mutation interacts with this mutation, and therefore you're okay. The reason why this is crucial is because the number of interactions between different mutations is astronomically larger than the number of actual mutations. So you can say, okay, we can throw away a big part of this, and we have some ideas of how to do this, and if we're being very liberal of throwing away these mutations from our genome and saying, okay, this one will never matter, we have about 10,000 mutations or 10,000 differences between two human genomes that actually are important. That is still 50 million interactions. That is a lot of interactions to study. Even if you being extremely liberal of which mutations you say don't matter to disease or don't matter to health at all, right? Still, you will never be able to, from general considerations, understand which interactions are possible, which interactions are impossible within these differences between human beings. People do try to study interactions of different things. This is a map of the interactions between different proteins of all the proteins contained within one particular virus. You can start to make predictions that, well, it will affect people differently depending on what kind of changes will happen in these proteins in the human. You don't need to look at the interaction of all possible things within the viral genomes. You can only look at the interactions of things that are interacting within this map. This is the good news. The bad news is that we're still very bad at actually making these maps. And another problem is that if you make a map of interacting of all human proteins, this is what you get. It's completely incomprehensible. And this is an interaction map of a very, very small number of proteins within the human genome. There are other things in the human genome that are very important. There are about 25,000 protein coding genes in the human genome that make about, let's say, more than 150,000 different proteins. There are also about the same number of different RNA molecules. Then there are also many different types of just regions within DNA that control the production of the RNA molecules as well as different proteins. So the genome is so complex, and we understand so little about the function of its individual regions without any interactions, that as soon as you add interaction to this picture, we just are completely lost. And if, and again, this is a big if, I'm not sure that this is actually important, but if interactions are important for medicine, we are so much farther away from personalized medicine than we are told in sort of newspapers and by optimists. So the only real way to go forward in this is to advance our fundamental understanding of cellular biology, really. And this is sort of the end of my talk and the end of my argument. If we fail to understand this, we will really fail to understand many of the implications of disease mutations, because a particular mutation only makes sense within the context of the entire set of changes in the genome and not just within the set of itself.